So that's a bit of a difficult task for me because this is not the usual crowd. So I'll try to strike right away with an example. So please pay careful attention to what's going to happen on the screen. Something will be flashed, and the aim for you is to try to recognize it. Okay, so that was 6, as you could tell. This is astonishingly boring kind of visual event for you. But for cognitive neuroscientists like me and like my peers, this is actually something which is really marvelous what just happened, because a simple light deflection onto your retina triggered a neural response in the back of your retina, which was propagated in the back of the brain and ultimately led you to believe that a 6 was presented on the computer screen. And this is all the more marvelous because we can actually look at the actual brain response to this kind of visual stimulation using a variety of techniques to try to see which brain area gets activated at each moment of time. So what you see here is the back of the brain. We flash the number 6 to the front of the subjects, obviously. And then we can identify within each milliseconds where the brain starts to be recruited. What we'll see is that around 60 to 80 milliseconds after the onset of the stimulus onto your retina, the back of the brain will start to light up. This is the areas which are known to process vision. This is known as the visual cortex. And from then on, within very few dozens of milliseconds, we'll see a very fast propagation of these early visual areas to what we call the ventral and the dorsal stream. These are hierarchical neural networks that transform raw visual input into abstract concepts. And we can see that this propagation occurs extremely rapidly within the first 150 milliseconds after the onset of this simple digit onto your retina, at least the retina of my subjects in this case. If we wait a bit more, we'll see that the activity is pretty much distributed across a variety of different brain regions. Around 350 milliseconds, we'll see that the activity even reaches the prefrontal areas, prefrontal cortex, which is known to host high-level cognition, generally associated with intelligent behavior. And if we wait even a little bit more, around 600 milliseconds, we'll also see activity in what we call the primary motor and sensory areas, which make sense in our task because we actually ask subjects to tell us what they saw on the computer screen by pressing a little button. Okay? So we have this super cool tool, Neuroimaging, which allows us to unravel both in time, space, within each subject, what goes on in the brain. But this is a pretty complicated signal. This is a very dense neural time series. And the whole goal for my research, and the whole goal, in fact, for many of us in cognitive neuroscience, is to try to develop experiments and tools and models to understand what this complex neural time series actually corresponds to. And more specifically, what we really want to do is to parse these signals in order to identify the series of computation that they correspond to, the series of computation that ultimately lead to intelligent behavior. In other words, what we're trying to do is to infer from the brain activity the algorithm of cognition, the sequence of operations that corresponds to cognitive abilities. Okay, so in my specific little research, within this sort of general goal, I also try to address 3 types of challenges. The first challenge is that I personally am interested in human cognition, cognitive abilities that are specific to our species, potentially as compared to other species. I think here of language processing, for instance, but also very efficient learning, one-shot generalization, and so forth. And for many of these cognitive abilities, we want to have tools that work at the single trial level. If you present the very same sentence over and over again to the subject, it's unlikely that the brain activity that corresponds to this sentence will be interesting. And we also want to have tools that scale to complex and natural tasks. And this we'll see in the second part of the talk. Most of the time what we do in cognitive neuroscience is to develop tools that work within very carefully controlled factorial experiments. And obviously, what we really want to go towards is tools that work to tasks that are potentially much more difficult, the kind of tasks that are interesting in AI and in machine learning. So what I'll try to show you within the next 40 minutes is a couple of studies that illustrates how we address this kind of challenges in the lab, starting from reading individual characters on the computer screen, and moving on to reading words and sentences. The goal, again, for all of these studies is to identify the sequence of operation that our brain implements to transform raw visual input into symbolic representations or categorical representations. But first, I'll start with some definitions and methods, which for those of you who work in cognitive neuroscience may be a bit boring, but just sort of to introduce the scene. So when I speak of brain activity, in this talk, I will be mainly referring to brain activity recorded with a technique which is known as magnetoencephalography. This is a type of scanner that we use, a magnetoencephalography scanner, or MEG. Most of the volume of this scanner actually is just a cooling system to work close to absolute zero temperature, because we have sensors, here magnetometers, that are located very close or relatively close to a subject's head, which do all the work. These are sensors which can pick up the fluctuations of magnetic fields elicited by neuronal activity. So when your neurons are active, they generate electric current. When you have an electric current, there is an associated electric and magnetic field. And because some of your neurons in the brain are aligned or spatially organized in a very specific way, their electric and magnetic field can actually sum up over space and can be picked up by distant magnetometers. So this is a technique which works extremely efficiently in the time domain. It's generally sampled around 1 kilohertz. So we have a snapshot of brain activity approximately every millisecond. But the activity is pretty difficult to analyze because it's in what we call sensor space. So we just measure the fluctuation of the MEG field outside the head. And there is a whole literature on trying to invert these signals to try to infer, given the sensor signal, where in the brain was the electric current most likely to have occurred. And I won't be speaking about this research today. I will just smoothly change or swap between sensor and brain space, depending on what I want to focus on. Okay. So this is for the brain activity. What about the algorithm? So algorithms can be defined in a number of ways. The traditional definition is a sequence of operation. For the purpose of this talk, we'll describe algorithm using 3 constituents: the architecture, the computation, and the representation. And to try to illustrate what I mean by this, I'll take an example. Here is an algorithm that implements the Fibonacci sequence, which, as you know, consists in just adding a successive term in a series. So, for instance, you start with either 0 and 1 or 1 and 1, and then you add these two. You get 1 and 1 equals 2, 2 plus 1 equals 3, 3 plus 2 equals 5, 5 plus 3 equals 8, and so on and so forth. You have a number of different algorithms that can actually instantiate this mathematical series. And this is just an example of such algorithm here in Python. And this is the very same algorithm now depicted as a little circuit, which will help isolate these 3 constituents that I mentioned to you before. So what do we have? We have 2 variables. At each moment of time, we hold 2 numbers. In this case, 2 integers that are called f0 and f1. These are the representation. This is the information that we hold at each moment of time. We also have a key computation in this algorithm, which is the plus. We just sum these two integers. And finally, we have the architecture of the algorithm. This is sort of the flow or the edges in the circuit of information within the algorithm that tells us where to move different representation at each moment of time. So the whole game for us is to try to decompose brain activity into this kind of constituent, to try to infer the computational architecture and to try to track the representation that are being generated by the brain at each moment of time in order to ultimately infer an algorithmic organization of brain activity. If you have any questions, feel free to interrupt. Okay, so in this first study that I've done together with Gabriel Mead, who is now a PhD student at UCSD, and Stan Dehaan, my former PhD advisor, we were interested in reading individual characters. And what I'll show you, the result that we get in the study is that we can infer the architecture that corresponds to neural activity, which is a heavily feed-forward architecture. And we can isolate computational properties within this architecture. And specifically, we show that the likelihood and the prior information is systematically computed within each of the processing stages of this architecture. And this is a bit dense, but I'll unpack this finding. So what we typically do in the lab is we take subjects to the MEG scanner, we flash letters and digits on a computer screen in the same way that I've done at the beginning of the talk. We record their brain activity, and we get these pretty complex time series that are defined both in time, but also in the 3 dimensions of space, and also in the dimensions of repetition, because we do these many, many times in a given subject. So for instance, here we have around 2,000 trials, 2,000 times we flashed a stimulus onto the subject's retina. And that's a pretty dense signal to analyze. So one thing that we can do to try to summarize the information that we are dealing with is to use so-called decoding analysis. So decoding analysis consists generally in fitting a very simple model, generally a linear model. And in this case, it's a logistic regression, to fit a classifier to predict what? To predict the presence of the stimulus given the brain activity, or given the MEG signals that we pick up at a given time point relative to the onset of the stimulus. So what do we have? We have, for instance, 2,000 repetitions. So we have 2,000 presentation of the stimulus on the computer screen. And we time lock the MEG recording with regard to the onset of the stimulus to the stimulus. We look at this MEG pattern and we fit a classifier to try to say, given this MEG pattern, was it more likely that there was a stimulus, or was it more likely that there was not a stimulus? So if we fit a classifier at, let's say, at 100 milliseconds after stimulus onset, we have a hyperplane across our different sensors or different voxels in the brain, and we get an accuracy or an area under the curve of 98%. So pretty much on every single trials, we can predict, indeed, accurately that there was a stimulus given the brain activity. And that's using a simple classifier trained at a unique time sample. We can fit another classifier at a different time sample, for instance, at 150 milliseconds after stimulus onset. So this is a little bit further in time. So if you recall, the activity has propagated now in the ventral and dorsal stream. And we can see that we can only decode now 74%, so accuracy sort of drops. And if we do this at a different moment in time, we get a different score. And if we do this systematically, we end up with a decoding score that varies over time. So on the x-axis is time, on the y-axis is our ability to decode the presence of the stimulus given the brain activity. And we can track the information which is available at each time instant after the onset of the stimulus, here at T0. Does that make sense? Okay, no feedback.
How are you defining sensor 1 and sensor 2? I mean, this is
in this case,
a projection of a very high-dimensional
exactly, yeah.
space.
In this case, it's actually the MEG sensor. So we have 3 or 6 sensors. And because it's a linear model, it doesn't actually matter which sensor is which in this case. Okay, so the purpose of decoding analysis from my point of view, we don't all share the same feeling, is essentially to summarize the amount of information which is available, in this case, at a given time sample. But it's generally insufficient to characterize a computational architecture that underlies the signal. And to try to illustrate this, let me show you 2 schematic architecture which potentially will be compatible with this decoding score that varies over time. So what we have in terms of an empirical finding, we have just something that tells us that there is activity for pretty long amount of time after the stimulus onset that we can decode. But presumably you can think of different architectures, either a very shallow sustained network that just maintain information within a unique processing stage, or something which is a long feed-forward network where information is only transiently coded at each processing stage, but continuously changes as a function of time. these two architectures are compatible with this decoding score over time because you can fit a classifier at a given time sample, retrieve information at that time sample, fit another classifier at another time sample, find information as well, and so on and so forth. So the way by which you can dissociate this kind of architectures is by doing generalization over time. So you not only train your classifier relative to the onset of the stimulus, let's say at time t, but you also assess the extent to which a given classifier can also decode information given other time samples. So for instance, you train here your classifier W2 on data which is only coming from, let's say, 200 milliseconds after stimulus onset, but you test the ability of that classifier to predict data which actually come from a different distribution, from a distribution picked 100 milliseconds after stimulus onset. So why do we do this? We do this because if you do it systematically, you end up with vastly different types of matrices. So here on the y-axis, you have the time at which you train your classifier, and the test axis is the time at which you tested your classifier. So the diagonal corresponds to what I showed you before. It's just a decoding curve over time, and the diagonal here would be identical. But the matrices are very different because the information is not propagated in the same way in the two networks. So if we take, for instance, an early decoder, in the feed-forward network, the early decoder will pick up activity in an early processing stage. That early processing stage is only transiently coding for information. And so your classifier, on average, only decode information for a transient amount of time. Whereas if you do it in this kind of network, the classifier that you trained at an early processing stage would actually be able to generalize at a later stage because the activity or the information has not changed, is still present in the same, with the same spatial configuration. And this is a case throughout, right? So you can fit another classifier later on. This classifier will pick up a different processing stage. This processing stage is transient. The classifier does not generalize, whereas in this case, it does. Okay, so you have these two matrices, which can be thought of as distances in a phase space. And so if you just replot the very same matrix now using an embedding to try to summarize these matrices, what you can observe is that under some assumption, you can retrieve the underlying architecture. So what you see here, each dot corresponds to a decoder, to a classifier, and you just plot the classifiers more distantly if they don't generalize with one another, and more closely if they do generalize with one another. So for instance, here, all of the classifiers tend to generalize along this timeline, so they will tend to be plotted in the same region, whereas here, the classifiers only generalize in a proximal fashion, so you end up with a sort of a long chain. Yes?
Even on the right-hand side, you could still observe something like that, but have a recursive structure in terms of which neurons are implicated. Like it could be the same neurons activated over and over again, but with different distributions. Yeah. So, yeah. Yeah,
I agree. So this is why here I wrote estimated state space as opposed to neuronal architecture, which I think is I think you point towards a very important subtlety, which is that you can instantiate using the same neurons. You can actually instantiate different processing stages by just changing the combinations of neurons, basically. Okay, so we call this temporal generalization. So how does it look in practice? So we have this fast feed-forward response from the back of the brain and then more interiorly. When we plot this data, or when we analyze it with a temporal generalization, we end up with this matrix of training time on the y-axis and testing time on the x-axis. When it's red, it means that we can significantly decode or generalize using that particular classifier. And what you see is that overall, you have a very neat and relatively thin diagonal early on, followed by an anti-generalization, which I won't be discussing today. And then it becomes much more large in terms of generalization abilities. Plotting the very same matrix now as using the this is just a plotting trick, there's no analysis there. You can see that each dot corresponds to a classifier, to a decoder. The edges correspond to the classifier that significantly generalize with one another. And then we can just animate it as a function of time. such that we can see where information is in that architecture. But I agree with you that this does not correspond to brain area per se. It corresponds rather to a state space, which I would argue is actually more interesting. Okay, so what we did then is actually divide the prediction of each decoder into prior, posterior, and sensory evidence, and to try to unpack this kind of idea. Let me show you a very classic illusion that we use in psychology to convey the notion that perception is not just a reproduction of what arrives onto a retina, it's actually an inference of that information. So here is a picture of a concert, which is very difficult to localize and analyze. Here is a slightly easier picture of a concert. Those of you who are big fans will recognize Beyoncé doing her favorite move. Here is another more anonymized concert. And here is another picture, which most of you would think is also a picture of a concert, although you would be completely wrong. This has nothing to do with a concert. It's simply a machine in a cotton field taken at night. And the reason why we can be so wrong at doing this estimation, again, is what I showed you at the beginning, is that perception is not just a reproduction of what arrives onto your retina, it's an inference over that, and this inference is using indeed what arrives onto your retina, but also using your prior expectations. Okay, this kind of illusion is true in the context of natural phenomena. It's also true in very minimalistic design. This is a very classic illusion where the very same symbol can be thought of as a B or as a number 13, depending on the context that you want to favor. So the whole game for us was to try to disentangle these three key computational constituents of perception in our task. So what we did is not just flash the stimulus as I showed you before. It's actually flash the stimulus and manipulate its visibility using a paradigm that we call backward masking, or that is called backward masking. So it consists in flashing the stimulus very briefly and then showing on the computer screen irrelevant stuff, here irrelevant letters, which will lead you to, will change the visibility of the stimulus. That's what we observe empirically. So the task for you is the same, it's just to recognize what is flashed here. But this mask will make it more difficult for you to see. So here is an example. At each trial there is actually a digit, I think it's always a number 6, but I'm not sure, and a mask. And depending on the latency that separates the digits and the mask, it's more or less difficult to see the target. So I'm super trained to this, so I tend to, I should have changed this slide, but I see the stimulus every time because I've done this task way too many times. But for some of you, it may be difficult, but after a while, you get used to it and you can start to detect when the stimulus actually appeared. Do some of you see the stimulus? Can you raise your hand for those who see it? Oh, cool. Well, maybe you don't need training. Okay, so what you see empirically is this kind of behavior. We change the visibility of the stimulus by changing the delay that separates the target and the mask. 0 is just catch trials, so there is no stimulus. And we collect subjects' visibility reports using a rating from 1 to 4. 1 being I did not see anything, and 2, 3, 4 is I sort of see something and I saw something. And you can see that they even have false alarms. So when there is no stimulus, they tend to say that they see the stimulus. Now, how do we modify the last element that I mentioned before, the prior? Well, we can control for the context, the previous trial that were presented. So if you expect the stimulus to be present, you're more likely to rate the current stimulus as visible. If you expect the stimulus to be absent, you tend to rate the stimulus to be less visible. So we have our 3 variables that we can now use to parse or to distinguish the decoding score that we were analyzing before. All right, so this is where we were. And now instead of looking at the extent to which we can decode information in the brain, we can see the extent to which this decoding prediction co-varies with our 3 features. So what I showed you here, this matrix is actually the matrix that corresponds to the longest delay. So this is 83 milliseconds between the target and the mask. But we also have shorter delays, 67, 50, 33, and so on. And so we can then correlate the extent to which these decoding predictions varies with the delay. And this is the result that we have. It's pretty much exactly the same as what I showed you before in terms of dynamics. And now the little red bubble indicates that there is a correlation between what the decoder actually predicts and the delay that separates the target and the mask. If we do the same analysis now with prior, so the extent to which subject expected the stimulus to be present or not, we have a different dynamic. You'll see that we have only the deepest part of the architecture, quote unquote, which is correlated with this effect. And this is true before the stimulus onset, so when it's before T0, and it's true throughout the trial. And now, if we look at the posterior, so the decision of the subjects, whether they actually rated the stimulus to be seen or not, it looks very similar to the evidence, but when we look carefully, we'll see that it's actually very different. So here is a snapshot when we combine the three together, and this is representative, so this is what happens generally. So we have the deepest part of the architecture, which seems to co-vary with the context. And then we have a ramping signal, which goes from probably something which corresponds to the early visual regions, ramp up very rapidly. But what you see is that the head of this signal is red, and the tail is green. And this is true throughout, right? So the head is the extent to which the decoding prediction co-varies with the objective evidence. And green is when it co-varies with the subject's decision. So what seems to be happening is that each little processing stage here first correlates with what was objectively presented to the subject's retina, and rapidly after start to correlate with what people actually reported having perceived. And you can see this if you analyze the time series for each decoder. So this is on average the response time for each decoder. So you see that you have first a peak response in red, which corresponds to the correlation with the objective evidence, and subsequently you have a maintenance of the prediction, which actually correlates with the decision. So it seems that we have a hierarchy of little computers which all do a similar type of job. They receive an objective signal that we call a sensory evidence or likelihood signal, and they seem to combine those signal with prior information to then code for the decision of the subject. But I haven't really parsed really the variables. So I've shown you sort of the architecture and presumably some computation that goes on within each of these little modules. But I haven't really shown you what type of representations are being generated. And this is what we'll discuss in the second study that we've done together with Laura Williams, who is a PhD student at NYU. Okay, so in this study, we did something very similar. We flash letters and digits on a computer screen. For every trial, it goes actually a bit more slowly. We ask subjects to tell us what they saw. But we did the task in a way that's parametrically controlled for a number of things. So for instance, we asked subjects, we flashed onto the subject's retina a 5 in a given trial, and we asked them, okay, what did you see? And we collect their response, and they report seeing a 5. When we flash a 6, they tend to report seeing a 6. When we flash an 8, they tend to report seeing an 8. That's all expected. But what was interesting in the study is that we also manipulated individual bars. So for instance, we could flash something which was between a 5 and a 6 in a given trial. So something ambiguous. And what we observe is that subjects' responses tends to be categorical. So when you flash something which is in between a 5 and a 6, subjects tend to either report having perceived a 5 or having perceived a 6, but not something in between. You have off-diagonal terms. And this is true throughout the board, right? It tends to be organized in lines. Subjects tend to have categorical judgment onto these symbols. And so you can collapse all of these results into the same type of curve that I showed you before, a sigmoid curve, where you have the probability of reporting that particular stimulus, so for instance a 6, given the evidence in favor of that stimulus on the x-axis. Although what was actually presented onto the retina objectively was something which was linearly related to that evidence. So the whole game, again, is to try to understand the sequence of operation that the brain undergoes to transform this linearly manipulated sensory input into a categorically judged output or behavior. So again, we use magnetoencephalography. This was actually collected at Neurospin thanks to Stan Dehaan. And we do the same type of analysis that I showed you before. So for instance, we fit a classifier, now not to predict whether there is a stimulus or not, but to predict the category of the stimulus. So whether it was a letter or whether it was a digit. If we do so, we can have a probabilistic output of our classifier if we use, for instance, a logistic regression. So we end up with this kind of results. On the y-axis, we have the probability of being a letter given the brain activity, given the MEG data. And on the x-axis now, we have the actual level of ambiguity. So in blue, we would have a digit, like a 4, and in red, we would have a letter, like an H. And this is at just after stimulus onset, so 30 milliseconds after stimulus onset, so there is no brain response in the back of the brain quite yet. But we can do this analysis at every moment of time, and we see that the line starts to bend around 150 or 200 milliseconds after the stimulus onset. So I'm showing you the same data now as a time series. So where on the x-axis is time, on the y-axis is still the probability of the classifier that is being output. And the shaded region is the moment at which we actually flashed the symbol onto the subject's retina. And we see that you have a ramping curve, which is linearly related to the evidence in favor of letters and digits. And if we wait a bit more, the line actually becomes more categorical. You see it looks like a sigmoid, at least statistically, it's a bit noisy, suggesting that subjects have now categorized in the head the representation of the symbol that was presented onto them. So this kind of analysis suggests that we can track the generation of a rather abstract representation, in this case, letters and digits, at each moment of time. But we haven't done this only for the category of the stimulus, but for a number of features. So here is the summary of this decoding score. X-axis is still time. T0 is the moment at which we flash the stimulus. And we vary the position of the stimulus, so we can decode that pretty early on. We also vary the identity, of course, the category, which as mentioned before, the ambiguity, so the level of with which it was difficult to recognize a letter or a digit. And finally, we collected subjects' responses, so they pressed the button to tell us what they saw. And we have a very nice cascade of decoding scores suggesting that the brain generates these different representations at different moments of time. Now, I should emphasize that this cascade is much, much longer than the recruitment of the hierarchical network of the visual pathways. I mentioned to you at the very beginning that when the activity starts from the back of the brain and reaches the end of the hierarchical network, we are pretty much around here in the time domain. So within the first 200 milliseconds, the whole brain is pretty much recruited. Whereas this generation of increasingly complex representation unfolds over a longer time scale, suggesting that it is perhaps the same neurons, but certainly a different combinations of neurons that generate these more complex features.
How you can identify the identity and not the category? Once you know the identity of the stimulus, you should know also the category, right?
Yeah, so let's think of like an abstract system that does not know anything about letters and digits. Like you fit a model on MNIST, for instance. Imagine MNIST with letters and digits as opposed to just digits. Nothing tells the algorithm what is a letter and what is a digit, right? This is a more abstract kind of concept which categorize, okay, all of these symbols correspond to digits and all of these symbols correspond to letters. So you can fit a classifier to recognize individual digits. So that's one type of classifier. And then you can fit a classifier that recognizes amongst all of these which are letters and which are digits. It may be not a super intuitive thing. It's just empirically we see that the curve dissociates. So I agree with you that it was not necessarily, it didn't have to be the case in this way. Any other questions? No. Okay, so again, these are decoding scores that vary as a function of time. And so it doesn't really tell us about the underlying architecture. And we can think of a bunch of different architectures. Let's take very simple cases just for the sake of the argument. Here is a feed-forward network where we add recurrent connection within each processing stage solely to maintain information. So there is no transformation of a representation via recurrence. So the stimulus goes into, let's say, the first layer of your little model, and that layer maintains this information over time. This information propagates to the second layer. This layer also maintains information over time, and so on and so forth, such that by the end of your trial, you indeed have information in your system, but it's distributed across different processing stages. Now, this is a different architecture than the following one, where you have no recurrent connection, but where you have the identity or the same representation that are being propagated across layers. So the beginning is the same. You input information to your first layer. This layer with a nonlinear transformation will generate a new representation to the second layer. But now you also propagate your identity, like in ResNet or in DenseNet. You propagate your identity, so you have sort of an incremental combinations of representation, such that by the end of your trial, now you have all of your information, which none of it is sustained in any processing stage, but all of it is nonetheless accessible in the deepest part of your architecture. Now, these are extreme cases, and what I'll show you is that we can parse the brain activity to try to recover this kind of dynamics. So how do we do it? Well, we do the same trick as before, so we do temporal generalization, but now for each levels of representation. So we have a time generalization matrix for the position of the stimulus, but also for the identity, the category of the stimulus, the ambiguity, the motor response. And what do? We'll just combine these different time gen matrices. We end up with a rather dense plot to look at, and nonetheless very beautiful, where each line corresponds to a decoder, and each column corresponds to a time sample relative to stimulus onset. And each of those lines that I showed you before is just a slice of that matrix. So we just show when each decoder trained at a given time point, when does it generalize, and what type of information it can actually pick up. So what do we see? We see that the early classifiers, they can decode the position of the stimulus for a relatively short period, and nonetheless above chance for something like 500 milliseconds. So it seems to be sustained, whereas the deepest or the classifiers that are trained much later in time, they can actually retrieve much more different types of representations and are also maintained over time. So we have something which is sort of in between the two extremes that I showed you before. There seems to be a recurrence activity, but they also seem to be an aggregation of hierarchical representations. Now this is even more obvious when we source localize these different effects. So here is a brain for each levels of representation, such that we can identify where and when information, different levels of representations are being available. And what do we see? We see that we have a very fast feed-forward response within the first 150 milliseconds. So the activity reaches the prefrontal cortex within the first 150 milliseconds, but only carries low-level information. There is not yet much going on in the other brains. And it's only after something like 200 milliseconds that you'll see seemingly recurrent, it's noisy, but it's nonetheless temporal lobe kind of activity, recurrent activity, which seems to build up the identity, the category, the ambiguity, and ultimately the response, which is propagated to the supplementary motor and motor areas. Okay, I'm running out of time. So this is all great, but a good criticism could be raised, which is that all of this only applies to carefully controlled factorial design kind of experiments, and it's unclear the extent to which we can apply this tool to more complex situations where it's difficult to a priori orthogonalize the identity, the category, the response given by the subjects, and so forth. So the whole game is to try to see whether we can develop methods to scale to more complex phenomena. So to try to illustrate that my optimism in this direction, as opposed to a strong conclusion, I'll show you very briefly one study where we investigate when we use these tools not to read individual letters or digits on a computer screen, but to actually read words that are flashed sequentially to generate sentences. So here is a recording of 100 subjects who went into the MEG scanners while they read sentences for about an hour. And the task looks approximately like this. Pay close attention. This sentence is very interesting. Okay, so this is the kind of thing that people have been doing for an hour and a half in the scanner and try to understand what goes on. There are a few questions every now and then to make sure that they understand what they read. And then we can do the same type of analysis, right? We time lock their brain responses to each individual words, and we can try to decode a bunch of things. So for instance, we can try to decode each letter that compose the word. So here is the alphabet on the y-axis, and the first bar corresponds to the number of letters that we can decode. So this is pretty easy, but that's a big visual feature. But we can also decode individual letters, whether there was an A, a B, a D, and so forth. We can also decode slightly more abstract types of representation, such as part of speech. So this is the word frequency, the extent to which a word is rare or common in natural language. This is, for instance, whether the word was an adjective or not, whether it was an adverb or not, and so forth, using a simple one-hot encoded classification using a ridge regression of brain activity at each time sample. Okay, I would like to just emphasize one result that is still a work in progress with Charlotte Cocheteux, who is now an intern at Facebook, which is based on word embedding. So for those of you who don't do natural language processing, word embedding is one of the most common tricks used as a first step to any NLP type of analysis. And it consists in transforming words into vectors, which are very handy representations for DeepNet and anything which uses backprop. And the way by which you build a word embedding to its simplest case is the following. You take a big corpora, and you simply compute the co-occurrence matrix between each pair of words. So you, for instance, count the number of times the word queen is present when the word king is present in a sentence or in a paragraph. And you do this for all pairs of words. You have a very, very large ill-conditioned matrix, so you can compress it using standard linear algebra, such that you have now a rectangular matrix of many words by a few columns, let's say 50, 100, or maybe a few more columns, which condense most of the information in that co-occurrence matrix. And what people have observed is that this simple operation allows you to extract semantic properties sort of in a surprising fashion. And you can do, for instance, analogy. So if you take the vector that corresponds to king, one of my favorite words, you can see that you can subtract man and add woman, and the word that you are closest to is queen. Which is very nice. There are many things you cannot do with word embedding, but nonetheless, this kind of thing is, in my mind, very cool. So what we did, what we're doing with Charlotte is very simple. We take the word embedding of individual words, the words that were presented onto the subject's retina, and we fit different algorithm to have a different sort of baselines. And then we simply try to say whether this word embedding linearly correlates with brain activity. Linearly correlates in the sense that we have a linear regression that can predict brain activity given these word embeddings. So how does it look in practice? This is the result from 10 subjects. So we have here on the x-axis is time. T0 is a presentation of the word that we'll be trying to predict given its embedding. On the y-axis is the extent to which we can predict that word. So this is an R correlation score. And then we have 4 embeddings. 4 embeddings that were trained with different amount of data. So this is, in yellow, the embedding that was trained with only 2,000 words, which is very, very small for this kind of algorithms or kind of analysis. In orange is 5K words. In purple is 20K words, and so forth. And what you see is that not only can we find linear correspondence with word embedding in brain activity as a function of time, but we see that the brain all the more correlates with word embedding, that the word embedding has been trained with a large corpus. In other words, the better the word embedding is, the more it seems to correlate linearly with brain activity. And I'll end with this because I think this is one of the elements that suggests that perhaps we can find a direct mapping between the algorithms that are being built in machine learning and optimized by our careful tools and large amount of data, and the way by which the brain actually instantiates this kind of operation, the way by which the brain, in this case, transforms a word into a semantic representation. So with this, I'll thank my students and collaborators, in particular Gabriela Mead, with whom we did the study with the letters and digits of a mask, Laura Williams at NYU, with whom we did the ambiguous letters and digits, and Charlotte Cocheteux for this very last study. And thank you all for your attention.
Thank you. So you talked a lot about feed-forward mechanism. So I was wondering, what is your view about feedbacks that happen in the brain, and if your model can tell us something about it?
Okay, so so the classic view, perhaps for the audience, is that at least there are a number of people who have data that suggest strongly that the feed-forward is sufficient for a number of key visual processing. For instance, there are many electrophysiology studies in the monkey that shows that if you simply record the feed-forward response to a visual event, depending on where you record in the hierarchy, you will have more or less hierarchical features. So if you present a face onto the subject retina, the early visual regions will only, for instance, code for the position of the stimulus, whereas the deepest part of the hierarchy, still within the feed-forward response, will represent the identity, the orientation, the emotion of the face. What we see in this task is not exactly like this, right? Because the feed-forward goes approximately for 200 milliseconds, and what we see is that this cascade lasts for something like 700 milliseconds, right? It's a very, very slow type of response. So the only way by which you can instantiate this kind of very slow cascade is through recurrence and perhaps feedback connections. There is sort of no other way around it. Now this is probably going to change depending on the task. This is sort of my opinion. I don't have evidence for this, but the more difficult the stimulus is to recognize, and the more you will need to engage recurrent processing and feedback processing to transform the raw visual input into an abstract category. So the way I think of this is, to go perhaps a bit too far, is that each processing step pushes you towards in the right direction. So if you already have a lot of energy or a lot of information that sort of you have a non-ambiguous image that is presented to the retina, then the feed-forward will be sufficient to push you to the right category. Now, if you don't have enough energy or enough information, so you need to sort of make a decision, it will push you to the right direction, but not enough. So you'll need multiple paths to ultimately reach to the categorical attractor. So this is how I see things, but this is just my opinion.
Any other question? Well, actually, I have one. Now that you have multiple tasks, multiple training on multiple subjects, can you think of training the network or training the decoding in one task and try to apply it to a completely different task?
So completely different. What we are starting to do is from speech processing in the auditory domain to the visual and vice versa. So that's the same sentences but different tasks. If you have ideas on what other tasks we could generalize to, that would be interesting. I'm sure people like, for instance, Stan would be interested in showing that the syntactical processes that you use to process language would perhaps be similar to those that you use to process mathematical equations, which also are structured with an underlying syntax. So I'm sure you can do this kind of neat things. The question is how scalable this is. I'm much more interested in pushing in areas where we know we'll be able to scale very efficiently. Going across tasks is not that direction, I think.
I'm curious about the resolution of MEG. So, in particular, to which extent can you decode the identity of letters of words? So, what is the max decoding?
Oh, so these are normalized. Yeah. So this is very, very small effects. The position of the stimulus can be decoded at 100%. The category of the stimulus is, I think, the peak. So this peak here is at 57, where 50 is baseline and 100% would be maximum accuracy. So it's actually a very, very small signal to detect if you solely use one time sample. So if you use the simplest possible model. Now there are different things into your question, because there is a spatial resolution on the one hand, and there is our ability to decode information on the other hand. And so you can sort of decorrelate these two questions. You could potentially have something which is not very well resolved in space, where you don't really know where in the brain things are happening, and nonetheless have a very good decoding score because your model is sufficiently good to assemble the different sensors and time sample, and vice versa. You could have something which is very well localized in the brain because you have hundreds and hundreds of repetition and very clear baseline and comparison, but nonetheless each trial independently would carry a very small amount of information. So overall, these signals are noisy. This is the primary difficulty that we face, unlike most of the machine learning sort of standard data sets. The biggest issue here is noise. We have noise which is orders of magnitude larger than the signal. When you do NLP or object recognition in ImageNet or in MNIST, the noise is only the thing that you don't really control for, but potentially that you could account for a posteriori. In this case, this is not likely to be the case. We have noise that comes from outside the subject's head, from subject's heart, from subject's muscles that we do not record and which directly impact the signal.
Okay, if there are no more burning questions, I would like to thank again Jean-Remi. And before we thank him, I would like to remind you that there is a lunch that is going to be at the usual place at the entrance of 24 Rue Le Mans. Thanks again, Jean-Remi.